afternoon, good evening, depending on where you are. We're really so very excited to have you join us. My name is Durham Wong Rieger, and I am chair of the Council for Rare Diseases International, which is co-hosting this with the Canadian Organization for Rare Disorders, where I'm also president and CEO. So this is a great excitement for us and the first opportunity to host a workshop around advanced therapeutic products, really with a focus today on gene therapies. We've got a very full agenda, so we're going to move right into it directly. Just to put up maybe the slide, Ange, to start. And we won't have an opportunity to everybody introduce themselves, but I'll just give you the format of how we're going to start. We will start with a couple of, just some survey questions. They're not really quizzes, they're not really polls, but a survey questions. And then, great. And then what we'll do is we're going, and you can see here the overall purpose of it, to provide an introduction to cell and gene therapies, really to talk about the partnerships with industry in terms of bringing these products to markets, and also really to raise awareness capacity, hopefully to, at the end of it, to have some information tools that are useful to you. Next slide. Okay, we're not quite on presentation mode, eh? Yeah, thank you. So one of the things that we're very keen on is to make sure that we're able to do a little bit of quick assessment. We're going to look at the range of cell and gene therapies with a focus, as I say today, on gene therapies. And then we're going to have a simulation and an exercise that might help you go into understanding or to at least experiencing some of the challenges that patients, parents might have in terms of decisions around gene therapies, recognizing that across the globe, of course, the access to these therapies is highly, highly varied. So we're probably not going to get it right for everybody. Next slide, please. So this is hopefully some of the pre-work that you've been able to take a look at. It is not a harsh requirement, but just to give you a little bit of an introduction. Some of you will be way ahead of the game, will know all of this very well, and are involved in gene therapies for your own conditions. Some of you, this may be just a beginning on. So we just wanted to provide you with a broad overview. Next slide. So what we're going to do is start with a poll, and I'll do that in a second. We're going to then give you some feedback on that. Next slide. Yeah, and then we're going to have two amazing presentations. One from Dr. Elizabeth High, who is now at AskBio, but has a long career in gene therapies. And then we're going to have another presentation from Dr. Elise Aon, who is the Hospital for Sick Children here, who again is a globally renowned researcher clinician and very much involved in terms of helping to make gene therapies accessible. Next slide. We'll come back to the others in a moment. We've got the case examples that we're going to present to you, and then you're going to have a chance to do some exercises around it. So why don't we leave it here for the moment, and maybe if everybody can, again, let me quick reminder to everybody, if you're just coming on, if you can just mute your audio, please. If you're not speaking, we'll eliminate the background noise and eliminate the switching of the screens, and to turn off your video. Hopefully everybody can find their mute buttons and are able to do that. So without much further ado, what I'm going to do is we're just going to start with 3 poll questions. This is not a real quiz. It's just to kind of get you warmed up in terms of the issues there. What you can do is you'll see that the first question is really just a hair bit of a knowledge question. And can we get the first poll up? Okay, so here's which of the following is not true about gene therapy? You can only choose one answer. Most gene therapies are one-time treatments. A mutated gene may be replaced with a healthy copy. A new gene may be introduced into the body. Viruses are often used as vectors to insert healthy genes. Stem cell transplant replaces damaged cells with healthy ones, or all of the above are true, which is not true about gene therapies, or if you choose the last one, you will acknowledge that all of the above are true. There's one that's not true, and you may put in your answers now.
My polls aren't working, I don't think.
No, I don't see anything working. Is anything working on the poll? Is it not working?
I can't make it work. This is
Yes.
Kathy.
Ange,
Yeah, I don't think the poll is working.
oh,
Is that your answer, Jerome?
I don't know.
It works for me.
Works
Oh,
for me.
perfect. Okay.
Yeah, you need to answer all the questions and it works.
Oh, we have to do all the questions? Okay, so answer the first question. We'll do the second question. Thank you, Russell, to put that in there. Sorry, I guess the way it's set up. Okay, answer question number 2. Okay, can you slide it up for question number 2, Angela? Can we scroll? Oh, I guess we can scroll yourself. You can scroll yourself on the poll. Apologies, I guess it's all set up as one gigantic poll. I apologize. Poll, the second is really just an opinion question. Gene editing is a method of changing the DNA or genetic makeup of a living organism. Which uses of gene editing would you agree with? This is an opinion question. There's no right or wrong answer to it. So you may answer it, and hopefully you can choose as many as you wish. Increasing the nutritional yield of fruits and vegetables, eradicating insects that cause deadly diseases, curing a serious disease in humans, developing allergen-free foods, converting algae into clean biofuel, eliminating the cause of life-threatening disease in embryos. Which one of these, or which ones of these, would you agree with should could or could be used for gene editing? Let's choose those. Well, I guess I can answer it myself if I want to. Okay, and the third one is, which of the following do you find acceptable risks or trade-offs? Whoops, I've lost the poll.
My poll just disappeared without
Yes,
giving in all the answers.
Can we kind of come back to that poll? Here we go. Okay, so
Thank you.
one, so you may need to answer them again. I'm not real sure. The last one.
Unfortunately, it wiped out all the answers for some reason.
Okay, so sorry, we're going to do this very quickly or else we're going to run out of time. Okay, we just do it very quickly.
Again.
Yeah, just do it again very quickly. I apologize terribly. You guys are not really an audience. You're just our test case for the moment. So at some point we'll get this right and we'll bring in real participants. It's okay. The third one says, which of the following do you find acceptable risks or trade-offs? Again, this is an opinion poll. Which would you find acceptable? Transplanted gene stops functioning over time. That's a risk that you may get a transplant and may not continue to work over time. There's a toxic or an immune response to the viral vector. So it's a virus that inserts the gene. There could be a toxic or an immune response to it. That there's a lack of immune defense when the patient's stem cells are removed. So in some forms of gene therapy, you have to remove the stem cells and then alter them to reimplant them. But at that point, you do not necessarily have a strong immune response. There's a risk that the gene therapy does not produce sufficient protein to stop the disease. So it may actually only partially address the condition. There are adverse reactions to the injection at the pain site, pain, fever, swelling, abdominal distress. There is the risk of a tumor or cancer as a result of the gene therapy, and the gene therapy goes into the wrong cell. Which one of these would you be willing to accept as a risk or a trade-off? Okay, choose the ones that you want, would agree with, and submit. We'll give people a couple more seconds. We're going to run out of time if we don't. Okay, so Ange, can we just show the results? Hopefully. All right, we won't discuss them now, but we'll save them and hopefully be able to come back to. So all of the above are true is what people say. And it's a little bit of a trick question because the stem cell transplant is not strictly a gene therapy, but in fact, it could be considered. So I think most people got it mostly right. Next one is most people would, this is again, was just for your own preferences. Okay, curing serious disease in humans is the one that received the most votes. Interesting. The last one, of course, is the one that's so controversial, and that is, should we be altering embryos? And this was the case with the HIV in children. And the following then, which would you accept as risk? We'll come back to some of these. Certainly the least problematic, of course, is the injection pain sites. Okay, great. We'll close it, and really we won't have a chance to discuss it, but hopefully we'll be able to come back to it, because I do want to get immediately to our first presentation. We are so absolutely thrilled to have Dr. Catherine High, who is going to be speaking to us. She's from AskBio. I'm not going to give you her full bio here because I think it would just take far too long. She is absolutely one of the pioneers in gene therapy and has worked in many environments, and in fact has been in some of the companies that have produced our first gene therapies. In fact, one of the first gene therapies that was ever approved. And she has very kindly put together an overview of an introduction to gene therapy. So Dr. High, I'm going to turn it over to you right away.
Okay, well, thank you very much. And I'm glad to be with all of you here today. I'm going to see if I can get my slide presentation up and underway. Okay, do you see it?
Perfect.
Okay, great. I'm going to start by echoing what you've already heard. That is that I was previously with Spark Therapeutics, and currently I have joined another gene therapy company. And I'm also on the board of directors of CRISPR Therapeutics, and I consult with Pfizer on gene therapy. So I'll just start by reminding you of something that you know very well, which is that most, although not all, rare diseases are caused by mutations in a single gene. And you know many of these diseases, cystic fibrosis, sickle cell anemia, muscular dystrophy, these are all caused by mutations in a single gene. And so this is a sort of US-centric view, but you can ascribe similar numbers throughout the globe. 25 million of America's 350 million citizens have a rare genetic disease. So in the aggregate, all of these rare diseases actually affect a fairly large number of people. And part of the goal of the Human Genome Project, which was begun in 1990, was to try to map and sequence all of the genes in humans with the goal of offering better treatment options for people born with serious genetic diseases. And of course, the goal was that this would be done through gene therapy. And in the end, it turned out to be more straightforward to sequence the human genome than to make gene therapy work. But so what is gene therapy and how does it work? So essentially, the most important slide that I have here today is something that describes what's called the central dogma of genetics. So the DNA in all of our cells is the same DNA. So the DNA in your liver cells is the same as the DNA in the cells in the retina, is the same as the DNA of the cells in your heart muscle. But what makes one tissue different from the other is which of these genes that are in every cell are actually brought forward and transcribed or read in that cell. So in the heart cell, the transcription apparatus is expressing heart-specific genes. In the retina, it's expressing retina-specific genes, and so on. So the RNA is distinct for every cell. The DNA is the same in every cell. And then this RNA is translated into a protein, and the proteins are really the action molecules in the cell. So here's the important thing. The DNA, which constitutes the instructions, is very long-lived. The action molecules, on the other hand, the RNA and the protein, are relatively short-lived with half-lives typically in hours. So the whole idea of gene therapy is that in order to make a correction, instead of giving the protein, which lasts for several hours, we'll try to give the DNA into the right cell, and that DNA, if the gene therapy works correctly, will last for the lifetime of the cell, and that cell will always be corrected and can then access the instructions that were not there when the person was born, but are there now and can drive the expression of the right protein. So, whoops, that's all right. So this slide is just, how do the letters of the DNA code translate to a specific meaning? So all of our DNA has only 4 different bases, or you can say letters, in it. A, G, T, and C. And those are shuffled in an endless array to spell out many different words. So that's the DNA. The DNA sequence is read in groups of 3. So it's called a triplet. They're read or transcribed in groups of 3 to produce the RNA. And then the RNA is translated into protein. And each 3 base word stands for a distinct amino acid. And these are the building blocks of the proteins shown here in little blue dots. So I'm going to try to give an example in words. So if we say that the DNA is composed of 4 bases, here we're actually using the 26 letters of the alphabet instead of only 4 bases, but you'll get the idea. So if the DNA is read as the cat was red and fat, actually there are lots of different 3-letter combinations that can make many different instructions, right? The cat was red and too fat. The cat was red and too fat for you. The cat was not fat. The cat had fun. The cat sat. The cat ate. So all of those are things that can be encoded in 3-letter words, okay? So that's really what your DNA is doing. So now let's look at how things can go wrong. So things can go wrong if there's a mutation. So this diagrams some things that can go wrong. So if the original sequence is shown here, and you can see it's made of A's, G's, T's, and C's, and each one of those would be read in a group of 3, right? So let's say it encoded the saying, the cat was red, in these nucleotides. So now let's say that instead of a T being faithfully copied as a T, it went in as a C. So now we would have something that said the cat Eos read. Okay, so that's doesn't quite make sense. It's not so far off. That might be a mild mutation. Now let's look at something else that can happen. A base can be stuck in, added, and that has the effect of throwing off everything that comes after it, because remember, we're always reading in groups of 3. So if we put in an extra letter, the cat was read, but now remember, we're reading in groups of 3, so it's going to sound like the cat duh. Okay, it doesn't make sense. So that's what can happen if an extra base gets inserted. And then, of course, there's a problem of what if a base gets deleted, as you see here. So then we might have the was read, but since it's read in groups of 3, you're going to have the azer ed. So, again, it doesn't make sense at all. So it's this combination of a relatively small change in the sequence can result in something that just doesn't make sense. And that's the crux of how mutations throw the reading frame off or result in something that's not really a word. And that's really the essence of what a mutation is. So how does gene therapy work to try to overcome that? And the idea here is pretty simple. For most of the gene therapies that are available right now, what scientists have done is harness the power of viruses. And viruses, as you may know, have evolved over many millions of years to attach to a cell and inject their own DNA into the cell. So here's the diagram of how a virus works. Here comes a virus. It bonds to a surface receptor on the cell. And then it comes into the cell, moves toward the nucleus, and deposits its DNA there. And that DNA starts driving the production of viral proteins. And those viral proteins and viral genes will assemble into new virions and make a spreading infection. So that's how a virus works. And the way that that's been harnessed to make gene therapy work is that scientists remove all of the viral genes and put in the therapeutic gene that they're trying to deliver. So in the case of Luxturna, it's a gene for a protein that's important for the function of the retina. And so instead of viral genes, this synthetic virus, which we call a vector, is not carrying viral genes, it's carrying a protein that you need in the retina. And when it's introduced into the right cells in the retina, it starts driving the production of the missing protein. It cannot make new copies of itself because it doesn't have any viral genes. And so that's basically how these vectors work. So you're using the outer shell of a virus, but inside you put a therapeutic gene, and this synthetic thing binds to the cell and goes into the nucleus and drives the production of the protein that's missing in the person. So this is just another diagram of what I just told you. So the DNA that you want to transfer is on a small structure called a plasmid, and it is packaged into something that on the outside looks like a virus. And that is loaded into a syringe and delivered to the correct cells in the person's body. And then now we have a cell that's been corrected. And that corrected cell can now function normally as if it had had that gene all along. So another way that we think about this sometimes is that the vector or the gene delivery component is like the envelope, and the gene is like the letter. So when you open the letter, you may keep the letter, but you don't want to keep the envelope. So essentially, this is what happens in your body. The DNA gets stabilized, but the envelope gets eventually degraded and, thrown away by the cell. So the delivery device is there temporarily, but the DNA is supposed to be long-term present in the cell. Okay, so any gene transfer strategy that anyone tells you about can be divided into 3 parts. The gene that's being transferred in, the vector that's being used to deliver it, so that's the envelope part. This is the letter part, this is the envelope part, and the tissue into which it's delivered. And these are just some of the many viruses that have been hijacked, if you will, to deliver therapeutic DNA. So there are lots of different types. There are mainly 2 types that we talk about for inherited disease. One is AAV. When AAV brings a piece of DNA into a cell, it mostly just leaves it outside the chromosomes. When lentiviral vector brings a piece of DNA into a cell, it actually integrates into the person's genome. I think I may have a picture of that later on. I'll show you. But again, the important principle here is that DNA lasts for the life of a cell, proteins have a short half-life. So we're trying to do something that as a treatment would only need to be given very, very infrequently. Actually, nobody knows how frequently it would need to be given to humans now. The longest data that we have for an AAV vector is about 10 years with observation ongoing. And for lentiviral vectors, about the same period of time. And these actually should last indefinitely, but time will tell. So I mentioned, so there are there are two, broadly speaking, two ways to get long-term expression. So let's look on the right first. This is a strategy that does rely on integrating vectors like lentiviral vectors. And the way this works is that the cells of interest, which are mostly cells from the bone marrow, are taken out of the patient, and the lentiviral vector is introduced in the lab, and then the bone marrow goes back into the patient. So all the gene transfer is taking place outside the body, and then the gene modified cells are now introduced back into the patient, and I'll show you a little bit more about that later. So that's called ex vivo. And why does it last for so long? Because this vector is now integrated into the chromosomes of the patient's cells, and every daughter cell that comes from the stem cell will carry that same piece of DNA, so you get long-term expression of the donated gene on that basis. In the other type of gene therapy, which is what we do with AAV, when an AAV vector goes into a cell, it does not integrate its DNA. It deposits it inside the nucleus, and it just sits there, separated from the normal chromosomes, but pumping out copies of RNA and protein. So this works to give long-term expression because we put it into long-lived what's called post-mitotic cells. So these are cells that are no longer dividing. And in that situation, we don't need integration because the cell itself is very long-lived. So we count on the fact that the cell lives for a long time. As long as we can get the DNA into the cell, it too will last for a long time, as long as the cell lives. Okay, so now I'm going to tell you a little bit about how these new gene therapy products are handled. So this is an AAV gene therapy for a very serious condition that children are born with called spinal muscular atrophy. So it comes to the hospital pharmacy frozen in a vial. So it's stored frozen. It goes to the hospital pharmacy, and it's held there in a freezer until it's ready for use. And then it's thawed and diluted. In the case of spinal muscular atrophy, it's intravenously infused. In the case of what Dr. Ayan is going to talk to you about, Luxturna, it's actually delivered to the operating room where the surgeon puts it into the right place in the eye. So in a way, this is like many other specialty pharmaceuticals. They're stored frozen, they're shipped to the pharmacy, and then they're given to the patient at the right time. Now, the other type of gene therapy, the one that goes into the bone marrow cells, is honestly a little more complicated to deliver. I'm always impressed with my colleagues who do this kind of work. But essentially what happens there is that the patient's cells first have to be harvested from their circulation. And then those cells are taken to a special laboratory, and the stem cells themselves are harvested. And then those stem cells have introduced into them the lentiviral vector that's carrying the extra gene. And then those cells undergo some further processing, and then they're put back into the patient after the patient has some additional conditioning regimen. So this is a therapy that pushes down all the blood cells that are in the patient so that these gene modified cells have a chance to take or engraft into the patient. So it's the patient's own cells, but now the cells have been modified to carry, in this instance, to carry a gene for hemoglobin, let's say in a person with either thalassemia or sickle cell disease. So it's a gene that corrects their blood disorder so that they can make normal red blood cells and no longer be dependent on getting regular transfusions of other people's red cells. They're making their own now. But you can see that's a little more complex than just thawing a vial of medicine and delivering it. So what is the current status of gene therapies for genetic disease? So I have to tell you that one of the most gratifying aspects of what I do is that every year I've been able to add something to this diagram. So the very first gene therapy that was approved in the United States or Europe was approved in 2012. It was for a very rare lipid disorder. And actually, it was conditionally approved by the European Medicines Agency. And in 2017, the sponsor allowed the approval to lapse. But it was a very important approval because it demonstrated that you could get approval of a gene therapy product. So this was now almost 10 years ago. And so gradually more gene therapies have been approved. This is for a rare form of immunodeficiency disorder. So babies born with this get frequent infections, they don't grow properly, and they used to die in infancy. And so now Strimvelis provides an avenue for long-term therapy for this. Luxturna, you're going to hear about from Dr. Ayan. This was the first AAV vector that was approved in both the United States and Europe. Zolgensma is for that spinal muscular atrophy, a very rare neuromuscular disorder, inherited disease that is also lethal in infancy if not treated. Zynteglo, this is a treatment for thalassemia. And Libmeldy was approved last year in the European Medicines Agency for a rare pediatric neurodegenerative disorder. Now, I will say that administration of this vector and of this one, now these are both these lentiviral vectors, are currently on hold because of the occurrence of an integrating event that led to leukemia for this one and for this one. They're on hold until they can investigate the cause and determine whether or not it was actually related to the treatment. And I mentioned to you that AAV vectors don't integrate. So these approved products, you know, that's not been reported for these approved products with AAV. Okay, so I'll say a little bit about how AAV vectors are manufactured, because that's what you're going to hear about later from Dr. Ayan. So this is actually a picture of a manufacturing suite, and you can see that everyone is gowned up in these bunny suits. We call them bunny suits, but anyway, it's because these are areas where there are very few, if any, particles in the air. So a great effort is made to make sure that nothing can get into the production system. And I won't go through all the steps in it, but essentially it's made in cells, and the cells are eventually split open, and the material is recovered from that. When it comes out of the cells, we have a mixture of particles that are holding DNA and particles that are empty. And so through various methods, it's possible to separate the fulls from the empties. And most products that go into patients are mostly these full capsids with the therapeutic DNA inside. So how is it delivered? We talked a little bit about this before. So this is actually a recombinant AAV being delivered into the retina. You can see here, you're looking through the operating room microscope. You can see that the surgeon is injecting into the back of the retina. It raises a bleb that you can see here. It's like a little bubble under the retina. 24 hours later, you can't see the bubble anymore. So that has to be given in a procedure done in the operating room that takes about 45 minutes. For spinal muscular atrophy, oops, misspelled word there. The condition that affects infants that is a neuromuscular disease, the vector is given by intravenous infusion. So again, this is mostly used for treatment of infants under about 4 months of age, and it's just infused intravenously. Okay. So what is the difference between gene therapy, cell therapy, and gene editing? So I tried to illustrate this in a diagram because gene therapy is approved for inherited retinal disorders. People are working hard to develop cell therapies for some of these retinal disorders, and there are trials underway using gene editing as well. So the retina is getting a lot of attention. So the gene therapy approach, the gene therapy is actually delivered through that procedure I showed you in the operating room to the right part of the retina. Now, suppose the person's retinal disease has advanced to the point where the cells that are the target for the gene therapy are not there anymore because they've progressively degenerated and they're not there anymore. Well, then that is a situation where people are trying to develop cell therapies. And the way they develop the cell therapy is that they take a donor cell and they grow sheets of these cells on a matrix that they can actually slide in a surgical procedure. So it's the same general kind of procedure, except that instead of a solution with vector in it, what they have is a sort of matrix platform that is populated with these cells, and that small sliver is slid into place here, and the RPE cells, the donated cells, start functioning. So that's a very important principle actually for many diseases. I mean, if there are still cells there that can be rescued, then you can use gene therapy. If the cells are all gone, the gene therapy won't work. And so instead, people are trying to design cell therapies. And that's true for a number of different disorders. Let's say Parkinson's disease, for example. So this is an adult neurodegenerative disorder that affects a specific part of the brain. If it's early enough, people try to give gene therapy. If it's more advanced, then they'll probably need to give cell therapy because the cells cannot be rescued anymore. And then gene editing, in gene editing, we'd have to go back to our diagram of normal and mutant genes. Well, let me see if I can very quickly do that. But what you're doing in gene editing is here. Essentially, in this type of situation, what the gene editing reagents can do is either, for example, they can cut out this edited base, or in a somewhat more complicated procedure, they can go to this spot where the wrong base is inserted and replace it with the right base. So that sounds like, science fiction, but it's actually already been done in a trial for thalassemia and sickle cell anemia. And what was done there was that a sequence was altered to take it out, and it allows the expression of a different globin gene. So that's the first example where it's actually been done, gene editing. Now, that was actually done, just so you know, that was done ex vivo. So in other words, the person's cells were taken out and then brought to the laboratory, and the gene editing machinery was introduced into their cells, restoring production of a normal globin gene and then put back into the patient. So that's already been done and reported out. People are working on a similar approach in a disease that affects the retina. So that's a trial that's also underway, again, attempting to correct the mutation in specific cells in the retina to restore vision. So gene editing is a much earlier stage. We don't have any approved gene editing products yet, but I expect we will in the next 5 years. So how do drugs go from an idea to an approved product? Most development begins with studies in animals, animal models of disease, and that's been true for a number of the gene therapies that are already approved. And then if studies look safe and as if they're working in animals, then human trials begin, human clinical trials. Clinical trials for gene therapy take place in phases. Phase 1 is typically a situation where investigators, physicians are trying doses that may begin with a low dose and then gradually increase to try to figure out what is the right dose for further study. And this is typically done for gene therapy in the intended patient population. So for some other classes of therapeutics like small molecule drugs, these studies may start in normal healthy volunteers. For gene therapy, they generally start in the intended patient population. For phase 2, typically this is just a larger study of what went on in phase 1. Typically, we take forward just one dose, sometimes 2 doses in a larger patient population. Which one is the best? Is it working? Now we start to measure efficacy endpoints. And then phase 3 is the last phase of testing before the drug is approved. The phase 3 study typically has a control arm that may be the standard of care. And this is the pivotal study that is submitted to the regulatory agencies to determine whether or not the drug will be approved and can be marketed. So for many rare genetic diseases, testing typically goes straight from phase 1 to phase 3. So in phase 1, you identify the dose that's going to be carried forward as the therapeutic dose, and then phase 3 is the pivotal trial. So how long does it take to develop a new gene therapy drug? I think that's an important question. So this is from a document called the FDA Summary Basis for Regulatory Action. Every time any drug is approved by the US FDA or by the European Medicines Agency, they produce a document that summarizes what was the evidence they looked at in order to approve the drug. And these are good reading. But they also typically in there publish a timeline of regulatory milestones. So I'll just show you that for Luxturna, which you're going to hear about from Professor Aon, the phase 1 study began. This is a jargon from the drug development empire, first patient in October 2007. And the drug, the phase 3 study began 5 years later in November of 2012. And the drug was approved by the US Food and Drug Administration in December of 2017. So 10 years in development. The phase 3 enrollment was completed in 12 months. Well, we don't need all that. Okay, now for Zolgensma, the drug for spinal muscular atrophy, the first phase 1 study, the investigational new drug application was submitted to the FDA in 2013, and the drug was approved in May of 2019. So you see the second approved AAV in the United States happened a little more quickly. So I do believe, I don't think I could come to work every day if I didn't, I do believe that the timelines for the development of gene therapy drugs will shorten. And we see that difference between the first one and the second one. And, you know, there's an old saying, the early bird gets the worm, but the second mouse gets the cheese. Sometimes, you know, if you're the first one doing something, then every time a new previously unsolved problem arises, you have to stop and solve the problem. But over time, you know, there becomes accumulated wisdom from the solving of these problems, and gene therapy drug development can progress a little more quickly. I think this is an important slide for me to show you. This is the number of investigational new drug applications for gene therapy products submitted to the US FDA every year beginning in 1990. And I think the important thing for you to see is what happened in 2017 and what happened in 2018. And what this reflects, and the number for 2019 was higher than the number for 2018. What this reflects is a dramatic expansion of activity in the gene therapy space. Will every one of these become an approved product? Well, probably not. We know that the approval rate for drugs that enter clinical testing is only about 20 to 30%. But it actually, as it turns out, is a little better in gene therapy than it is for many other classes of drugs like small molecules or monoclonal antibodies. So this is good news that there's a lot more activity in the space. All right, so anyway, I think I made that point. I'm just going to give you this example. There are no approved products for gene therapy for hemophilia. But look at all these trials that are going on. The ones on the top are for hemophilia B, the ones on the bottom for hemophilia A. And, one thing that I can say I've seen over a period of many years, when you start to see many trials for a single disease, it usually means that things are working. And that's why there's so much activity in the space. So sometimes people ask, well, there's so many different trials, which one should I go into? But basically, when you see a lot of trials for the same disease, that's good news. Okay, now I'm just going to close with one thing. This is actually a film that was made by a television station in Boston about the first child that received Luxturna after the drug was approved. And this is what I think is very important. I mean, as you may guess, in that 10-year interval when we were working on trying to get Luxturna through the drug development process, through the regulatory approval process, there were definitely times when it felt like, okay, maybe this is just a bridge too far. Maybe we're not going to get there. But in the year after the drug was approved, more people got it than in the 10 years of the drug development process. And so that's just, I would say, my homage to the people who actually market drugs. I mean, I think that what I learned in that was if the drug can get approved, they can get it to people. And, that of course is the objective of all the work that we're doing is, to get it out and get it to people. But that's the reason to try to get a drug approved is because it will reach many more people in a much shorter period of time than you can ever do.
It was my first day back in school and I was in math class and my teacher was writing on the board and when she finished writing, I was like, wow, I saw it. And I went home and I told my mom, I was like, Mom, I could see the whiteboard.
Homework on his own, he did his projects on his own, and he's never done that before.
To get pizza with my friend, it was like 7:30. It was like a little dim out. I've never rode my bike at all at night, and now I can. I'm gonna go so fast.
Okay. Well, thank you very much for your attention. And if we have time, I'm happy to take any questions.
Okay, so thank you so much, Dr. Hyde. That was probably the most comprehensive but comprehensible overview. I know you've got to leave, and what we're going to do is jiggle this schedule a little bit. Thanks to Dr. Aon, she can stay with us beyond this. So instead of waiting to take questions, we did have a few questions that came into the chat. Some of them are quite technical. I will say is that Dr. Hyde can only stay with us for about 5 more minutes. So if you have a question, put it into the chat. We'll try to get it to her. If you've got questions that we can't answer, we will stack them up and we will pester her afterwards to try to get an answer. How's that? So she's on call here. There was a very early question that said, which I thought was somewhat technical, from Hussein, says, could epigenetics play a role in cell and gene therapy? So instead of changing the DNA sequence, you would be changing whether a gene is expressed or not via methylation and or acetylation. So do we have a technology to do this? Kind of a technical question, but maybe worthwhile.
Well, I do think that there are efforts underway to alter gene expression through epigenetics. That's not what I work on, and I'm sorry I'm not completely up to date on where things are in that field, but I know that there's work going on to do that.
Great. There's another question that says, I'm not sure if it's a little bit later in context, from Rob. It says, you said delivered to the cell. How does that work? How does the transferred cell know what cell is the right one? How does it know where to go? So you're injecting it. How does it know where to go?
So this has to do with, actually, there's several different layers through which that process is controlled. So I'll tell you that the AAV vector that you're going to hear about from Professor Aon, that vector will attach to cell surface receptors on the retinal pigment epithelial cells, but it doesn't get into the cell layer right next to that, the photoreceptors, very efficiently. It just doesn't have the right cell surface receptors to pick it up very well. So that's one way that, so in a complex target tissue like the liver, for example, the AAV vectors, there are abundant cell surface receptors for them on the hepatocytes, but not, let's say, on the blood vessels. You can't get into the endothelial cells in the liver because they don't have the right receptors on the cell surface. So that's one way to control where it goes. Another way that people control where it goes is, even if it can get into the cells, if the promoter only works in a certain kind of cell, you won't get expression of the gene. Even if it went into another cell type, it won't express there because the promoter is cell type specific. So there are several layers of control to make sure that it only gets expressed where it should be.
That's brilliant. Thank you. There's a technical question here, which I'll try to shrink down a little bit. And this is for the, talking about the muscular dystrophy. This is one of the longest genes in the human body. What could you tell about micro or mini dystrophin using a smaller part of the gene in the body?
So this is a strategy that is not unique to muscular dystrophy. There are a large number of genes, not a large, actually about 90% of all gene sequences will fit into an AAV vector, but 10% of them will not. And dystrophin is one example of it's, I think, one of the longest genes in the body. Another one is factor VIII. This is the thing that's missing in hemophilia A. Those things don't fit into an AAV vector. And so what people have tried to do is make truncated or shrunk down versions of the gene that can do the same thing. And I think, this is a challenging area, but we are in the midst of a revolution in protein design. And there are many scientists who work on making more active versions of a protein, or they can shrink the protein down and try to make sure it will recover most, if not all, of the same function. So, when all of this work in gene therapy started, we just couldn't do things that were too big for an AAV vector, but now many of those are being approached.
Amazing. I'm going to ask you one other very quick question, which is a little bit beyond, which I think is a great one. Russell asked the question, previously as well. So we're talking about using, the AAV as a vector, but we're also using AAV to develop vaccines. Is there any problem with, having, an immune response already, an antibody to the AAV, if you're going to be, doing a vaccine, and then you've now got a gene therapy, it's going to be delivered by the same vector? Do we have a concern?
So I think that the vaccines, for example, for COVID, are mostly adenovirus. They're not AAV. So the only thing that we've been concerned about around COVID vaccines is that, as you know, if you've gotten one, you can feel unwell afterwards. And, because your body is making a brisk immune response to the COVID proteins, which is good. But you don't necessarily want to be exposed to a gene therapy vector in the same time frame when your whole immune system is sort of riled up trying to make a response to COVID. So, we try to separate in time getting a vaccine of any type, doesn't matter if it's just a flu vaccine, from getting an AAV vector. In other words, if you, in gene therapy for genetic disease, we want a minimum immune response. in any kind of vaccine delivery, you want to jazz up the immune response so that you'll have immunity to, whatever the thing is, flu, COVID, whatever. But no, they're not rule out. I see a question here, is the current COVID vaccine? No, it's not a rule out. You just don't want to be doing them at the same period of time.
Great question. So thank you. I know it's right at 9 o'clock and we promised that you would be liberated at the top of the hour. We cannot thank you enough for such an amazing presentation. And really, we've got it hopefully to be able to continue to use it. Thank you very, very much. And
Sure. Lawrence Willard, I see your question. I'll send you an answer.
okay, so she's promised that she will fill up the chat box with as many questions as you want. We will make sure we get them to her and we will be able to carry the responses back. Thank you so very much.
Bye-bye.
Okay, bye. All right, so we're going to pivot a little bit here. And really, we're going to have to change up our agenda a little bit. Dr. Hai was great in terms of having such a comprehensive presentation, a little bit longer than we had anticipated. So we're going to pivot over to Dr. Ayan, who's going to talk to us, as Dr. Hai indicated, more specifically around around Luxturna gene therapy for retinitis pigmentosa that you had allusions to already. So I think without further ado, we might move right into that. And Dr. Ayan is with us and has kindly agreed to stay with us to be able to take questions afterwards as well. So I think we will start the presentation there.
I'd like to show this picture of a beautiful patient of mine who's blind. Who would know? Blindness is invisible and in too many cases, silent. I'd like to bring you back to the 80s and the 90s when Jurassic Park was written and made into a best feature film. You may recall that it's the story of a graduate student who recreates a dinosaur from fossilized mosquito, leading to a wild park of extinct animals. That was science fiction then. The internet was barely around. At that time, too many patients were still told, don't drive, don't have babies, you're gonna go blind, and worse, nothing can be done. In those years, we knew of 2 or 3 retinal degeneration genes, whereas today we know over 250 genes. And today we're discussing gene therapy, which is quite marvelous. So what was thought to be impossible for us has now become possible. So what is retinal gene therapy? First, you need to have what we call a disease phenotype. So what does disease look like? What happens? What changes? What doesn't change? Then you need to have a genetic cause identified. And for that, you need access to genetic testing. If you're lucky and your cause is identified, there may be some animal models that exist or that can be created to recapitulate your disease, to better understand what's going wrong, and work towards developing treatment strategies that are then tested into clinical trials with the hope of improving the outcome. When we talk of inherited retinal degeneration, they all call it blindness. We refer to the degeneration of the photoreceptors, which are the light-sensing cells in the back of the eye. The retina, you have to see as a light-sensing tissue that totally lines the back of the eye. When the retinal degeneration occurs early, we refer to it as Leber's congenital amaurosis, and afterwards we refer either to retinitis pigmentosa or retinal degeneration. In clinic, we have a very useful tool that we refer to as the OCT, which gives us a cross-section of the retina. This allows us to see the different layers that host different type of cells, the photoreceptors being the layer here behind. This is a normal individual. On the right is a patient affected with LCA due to RPE65 variant at 25 years. And you can see how the structure of the retina is different. It is thinned and it lost many of the layers because the cells are dead. LCA and related diseases are rare diseases. So LCA is thought to be about 1 in 100,000, and there's many different genes that cause that. Today, we're going to focus on one subtype, is LCA, meaning early onset retinal degeneration, due to variants in the RPE65 gene. That accounts to about 8 to 10% of LCA, which may seem like a small portion, but it's actually a significant portion. In addition, we have to keep in mind that early onset retinal degeneration are the leading cause of childhood blindness. So what's the big deal with RP65? RP65 is actually a critical enzyme that allows the recycling of the vitamin A once the light hit the photoreceptor. And that is critical for the visual cycle. That's why we say eat your carrots. Without RP65, the benefit of your carrots just can't reach the retina. So luckily, in addition to knowing about the biochemistry of RP65 related disease, there were different naturally occurring mutant models of the RP65 mutations. And these were powerful tools to better understand what was going wrong in this condition, but also to develop different therapeutic approaches. So I'm presenting you here Lancelot, which is one of the naturally occurring mutant. He's a Briard dog. And initially Lancelot was blind, but now can catch the ball and bite his peers because he was treated with an injection of a RP65 gene packaged in a virus. In 2008, it was shown that this approach in dogs was safe. There was no adverse event, and there was visual function recovery. It was an unbelievable moment. So what does that mean for the patients? In humans carrying RP65 mutations, the visual problem usually starts before the age of 5. It's a decreased visual acuity, decreased ability to adapt to a dim environment. The eyes are usually shaky. We say they have nystagmus. The visual acuity decreases rapidly, hitting 2200, which is the level of legal blindness, and going even lower. The retinal function is measured with the electroretinogram, which is always severely abnormal. Consequently, the visual field, the peripheral field of the individual narrows, and by the teenage years, they're about 10 degrees, which is very small. In cases of low visual function, it's harder to measure change in a reproducible manner. The visual acuity doesn't become useful because in the low levels, there's more variability. The visual field's no longer useful. So for the RP65 clinical trial that was planned, in partnership with the FDA, they developed 2 outcome measures that would allow reproducible quantification of visual function and functional vision in these patients. So one is the multiluminance mobility test, or in short, MLMT. And the other one is the full threshold sensitivity, FST. We'll focus on the MLMT, which is a maze. It's a maze with a variable number of obstacles, paths, but that is ran in varied levels of luminance. So for example, 400 lux is like a bright fluorescent office setting, whereas 1 lux is the level of a moonless summer night. So when you have RP65, you don't see anything in these settings. So this is an example here of a patient with RP65 who is going to undergo the MLMT, and you're going to see how it's measured. So there's obstacles. So each obstacle has a value. There's also the speed at which the participant functions, and there's the number of errors. So these have all been validated and standardized. So you can see that she has difficulty making her way and is very hesitant. So I'll just stop here. Whoops, sorry. So what is this treatment? As I mentioned in what the dog received, it's the human RP65 product that is packaged in a virus in order to infect the cells under the retina. So the product is in a syringe, is injected through the eye under the retina to infect the pigment cells and restore the visual cycle by redelivering the missing enzyme. So this led to the phase 1 of the assessment of this treatment. So that's the first step where we want to assess toxicity of a product and determine which is the best dose. So they tried 3 doses and showed safety with all 3 doses, but more efficacy with the highest dose, which was then used in another cohort of patients and followed for a year. And then the other eye was treated and followed for 12 months. So this actually was also the first study where both eyes were treated. The summary of the phase 1, the most important thing is that the treatment is safe, that there was no serious adverse event related to the product, there was no significant immune reaction, the best dose was identified, and even though the MLMT was at its beginning, they were able to show some efficacy at that time. So this was used to refine the MLMT and then move forward to the phase 3, where you really assess the efficacy. So in the phase 3 trial, they use the higher dose that I mentioned, and they inject both eyes at a week or 2 interval. The control group is observed for a year, and then they cross over and they're treated. So everyone gets their treatment, but a third of the group is at 1 year interval. The endpoints in this study are the multiluminance mobility test and also visual field and the FST. So these are the results of the multiluminance mobility test, which shows, so on the vertical axis is the lux score, which means the change in luminosity that the individual can achieve, the change in lowering the luminosity. And so there's a green line here that shows that within a month, there's a rapid, steady response to the treatment where the individuals can easily navigate at the 4 lux level. The control group that's then treated at a year even show a better response, where 89% of that group was able to function at 1 lux at 1 year, and that remains sustained for 4 years. So that's quite extraordinary. So in terms of the MLMT, this is how it looks. So you can see that there's less hesitancy. There's also increased speed. And she doesn't fall for the obstacles. She actually has a perfect score. So needless to say, that was a very exciting moment. In terms of the FST assesses the residual retinal function, but in a very broad way. Like when the other tests are not sensitive enough, this is a different approach. And what was surprising is that if you look at the lower graph, which is the treatment group, there's improvement at all ages. And that's quite surprising because when you're in your 40s, the disease is usually well established, though the disease severity in this situation is not always related to age. So following the treatment, there was improvement in the rod function, which is the night vision, and to a lesser degree, but still an improvement in cone function in some individuals. There was no improvement in visual acuity, and we did not expect that. So in summary, the phase 3 trial showed that Luxturna is a safe treatment and shows durable, reproducible efficacy. There's improvement in mobility at lower light level, and there's an increase in the retinal sensitivity at lower light levels also. There was no significant immune reaction, and the adverse event observed were related to the procedures, which are standard, quote unquote, not related to the product. Importantly, what is to remember is that Luxturna is not a cure. It's a treatment, and it's a treatment that significantly impacts the quality of life of patients affected with RP65 related retinal degeneration. So what next? Well, we need to break the silence because there's not enough people talking about this situation. Since then, there's over 40 clinical trials that have been initiated and ongoing on different genetic subset of retinal degeneration. Ones that Luxturna was shown to be safe. Now everyone has to be redesigning their own genetic subtype. Unfortunately, it's not a copy-paste mechanism. Each genetic subtype has to be managed on its own. But the big difference in our field is now inherited retinal degeneration are actionable diseases, means something can be done, whereas before that was not the case. So what have we learned? We learned a lot of things. And I just wanted to go over a few of these with you, which may seem a bit odd, is that you need to understand the disease. Like for us, that means you need to define the phenotype, the different aspects of disease to see which one of these parameters is most sensitive to show you change. So the outcome measures were somewhat arbitrarily selected because there was no natural history for any of these retinal degeneration. So it's very important to go to follow-up examination, even though sometimes it's frustrating, because the longitudinal information obtained is critical to best interpret response to treatment. Where we also have been weak is understanding the impact of the disease on the patient's quality of life. So until recently, there was no patient-reported outcome measure specific to retinal degeneration, which is changing now, and we're working on establishing the pediatric version. Who should be treated, and how long do we have to treat? We have a while to treat, and it all depends on the expectation. So the phase 3 showed that even at the age of 40, you could have a significant visual improvement in terms of mobility and quality of life. But those are dilemmas. Do you treat the teenagers first? Because past the teenage years, you're at risk of losing more vision very fast, and then your ability to recover becomes a bit jeopardized. Or do you treat the very young and take some risks? We don't have all the answers, but groups are formed nationally and internationally to address this, to make sure that the best decisions are made. We're lucky here in Canada, we have a registry for patients with inherited retinal degeneration. It's called the Fighting Blindness Canada Patient Registry, in which patients with inherited retinal degeneration, and especially if there's a genetic result, the patients can be identified for any study pertaining to their condition. Access to genotyping was only available in Canada province-wide in the recent years. In Ontario, we were privileged to have this available for the longest time. So the number of patients eligible to such a treatment will increase in the next 2 years just because many patients have not yet been tested. Something that has to be discussed with the patients clearly is the expectation. So what does a treatment mean? Ask to 3 people around you and people are going to have different responses. So it's important to have that dialogue. So how do we get this to the patient? The FDA approved Luxturna in December of 2017, followed by the European Union. Health Canada approved only this last October and was approved by the provinces in the new year. And I understand that the dossier is under evaluation for reimbursement. So we could not administer a treatment without having affirmation that the province will reimburse for the treatment, because the hospital has to purchase the treatment and it's around $1 million for both eyes. Individuals who are eligible for this treatment are usually above the age of 4 years, need to have a viable retina and 2 mutations in RP65. What we've done in the meantime is that the Novartis, the company, set up Canadian treatment sites, because it's not a treatment that could just be administered in any clinic. And so there's 2 sites in Toronto, Sick Kids, where I am for the children, and Sunnybrook for the adults. And there's 2 sites in Montreal, also in Quebec, because the bulk of the patients are in Quebec and Ontario, but that does not mean that the other provinces will not have access to the treatment. The patients will be treated in those sites for now, as long as the home province accepts to reimburse. We form treatment teams, we've been trained, we've developed internal standard operating procedures. We have a national advisory board through which we have a meeting tonight where we develop national guidelines for patient selection and management, and how are we going to make this happen. So a lot is happening, even though we haven't been able to start. I'd like to close with a personal story of a patient I had in my clinic last fall, who was a new diagnosis of RP65. So I go, so my routine, difficult conversation. And then I ask if they have questions, and the little boy says, yes, I have a question. He says, what are you going to do for me? And usually we can go through the supportive measures, but this was different. I knew Health Canada was about to approve, I assumed Health Canada was about to approve Luxturna. And I could say, yeah, we can do something. I promise we'll do something, and hopefully it won't take too long. And I do hope that we can rescue and improve his visual acuity so he can improve adulthood as a more independent person. So in summary, what we thought was impossible is possible. And you need to remember and spread the word that inherited retinal diseases are actionable diseases, as gene-specific treatments are growing fast. But first, do no harm. We need to understand the disease, we need to understand the patients, and need to make sure we choose the right patient for the right treatment. And lastly, although Luxturna is a wonderful product, it's a treatment. It's a treatment that improves the quality of life of patients affected with RP65 disease, but it's not a cure. Thank you for your attention.
Thank you so very much. And we do very much appreciate the ability to apply that to an actual example. So Dr. Aon is with us, and we'll do a couple of things. Maybe if there are other questions and you would like to put them in the chat, we can ask her to respond to them. She's also agreed that she will stay with us until 10. So we're going to ask her to join one of the breakout groups and maybe come back and have more commentary later, because one of the exercises around it. But at the moment, maybe there is a question here for you. From Russell, it says, so if all ages improve with Luxturna, and this is a childhood onset disease, what is the cellular impact? Are all cells remaining viable throughout the life of the patient and therefore treatable?
Hi, Russell. when you inject the product under the retina, in order to get efficacy, the product has to go into the cell. And not all cells get transfected, meaning with one injection, you lift the retina, the fluid stays under the retina, but what we call the transfection rate, the ability, the quantity of cells that get transfected is not 100%. And it's not predictable. You'd like to predict that the more viable cells you have, the more transfection will happen. And also it depends where the product is in comparison to the amount of viable cells. So in retinal degeneration, you can have patches of cells that are dead. Those likely will not respond to the treatment. But if your injection is under a patch of more cells that are alive, the ability to respond will be greater. So there is variability in the treatment response. And to date, that is partly related to patient selection, technicalities about the injection, and there's some factors that we probably don't know. When individuals around the age of 40 show response, all it says is that they have viable cells. And we can determine that a priori by using the FST. So if we have electric recordable photoreceptor response, we know that there's viable cells. And that varies between individuals. Like there's some individuals have very poor, more severe disease early, and others are a little slower and later, and we don't have all the answers why. There's other genetic factors that are involved.
Thank you very much. So we don't have another question at the moment. I'm going to encourage people to put them in because we are kind of running tight on time. We're about, about 20 minutes behind where we want it to be, but it's all good. So what we're going to do is to actually put you into breakout groups, give you an exercise, and then ask you to talk about it in your breakout groups, and then we're going to ask you to report back. So in order to do that, I think you've all received a copy of the exercise ahead of time, but you probably only received it very shortly. Can we, I'll just pop up the scenarios for you now, and so you'll have it all together. And then I'm going to tell you what groups you're in, and then hopefully we'll be able to have you move into those groups. We also will have leads in each of the group that we're going to ask to maybe just host the discussion and then be able to report back. So we'll put up the exercise and then we'll let you know who the leads are, and then you'll go automatically, hopefully, into a breakout group. So the two exercises that you've got, you may choose one of the two to work on with your group, are quite similar. One is on the retinal disease that you just heard about. I won't read it in great detail because it is very much a, back up. Okay, it is very much a takeoff. It's not an exact replica of what is the real treatment. It is just a case study. It is around the delivery of a gene therapy through a viral vector. And that we have there, it says about 60% experience significantly improved vision and all some improvement. So this is for the patients that were in the clinical trial, and the data says that up to 4 years, the improvement remains with only minor adverse effects. What we do know is that there are 2 other clinical trials that are recruiting patients for the same disease. One is a gene therapy using a non-virus vector, and another one including younger children, both with potential for better vision outcomes. Again, this is just a scenario, so it's not necessarily real life. We've got 2 patients that you can consider. One is a 17-year-old who has severe IRD, and he's got and has been experiencing progressive decline. Another one is this 13-year-old who has moderate to severe, but he's been stabilized over the past 2 years. What we like you to do is just assume the role of a consultant to the patient, to the family, and to the physician. And what you want to do is to really consider what are the options for these 2 patients. They're different patients, whether or not they should try to apply and advocate for access to this gene therapy as soon as possible for 17-year-old, for the 13-year-old, whether they should wait and enroll in one of the current clinical trials that might actually lead to better outcomes using a different virus, or should they just wait and not do anything until maybe, again, with all the research that's going on in place, they should have another therapy or another clinical trials. And then the next one, and the next scenario, okay, is similar, only this is with the case that is with a neuromuscular disease, and the question will be very much the same. You've got, you know what, maybe we'll not do that. And what if we just do the one? I think just in the interest of time, I'm wondering if I should just have people focus on the one. Because we introduce a lot of complexities into the whole scenario and we don't have a whole lot of time. I was going to give you a choice of which scenario you wanted to do. Well, here, let me just do it anyway. Sorry to be so indecisive. Put it back up. Maybe people would prefer. We'll give them a choice. So this is, again, for those of you who may be aware of spinal muscular atrophy, this is a little bit of a takeoff onto that case where you've got to treat the children who are severe. They would only be able to be treated when they're under 2 years old or very small because of the amount of gene that you would need. What you've got is two different treatments. One is, in fact, a copy of the protein injected directly into the patient, so it's not a gene therapy. The protein that makes the necessary the protein that is necessary for muscle development is injected directly into the patient, but it has to be injected every 4 months, injected into the spinal cord. The second is a gene therapy. It is, in fact, injected in the body with the hope that it will continue to reproduce, then stimulate the gene, or it provides a copy gene. It will make the protein on a continuous basis. The question is, if you are looking at the children that are affected, you know, what do you want to do? Do you want to, if you're already started on the protein injection, to remain on that protein, transition to the gene therapy? If the child is under 6 months old and just diagnosed, what is the best strategy? Should they immediately go into the gene therapy, or should they actually start on the injection therapy, which has been shown to be effective? Okay, so we're going to move you into your groups, and we're going to ask then you to consider the cases and hopefully be able to come back to us with a report back. You may choose one of the two scenarios. You're going to be moved into your groups. I'm just going to read out who the leads are, and hopefully you're all still here with us. Ida, hopefully you can take on one of the leads. Kwani, one of the groups. Mary Akua, hopefully you take one of the groups. Monica, I think is still here. I'm not sure. I'm hoping that Monica is still here with us. It's very late, I know, in Australia right now. Nicola Horsfall, Rob Camp, Russell Weir, and Tanya Collins. So you'll all be moved into your groups. If those of you who are just listed as leads can actually help lead the exercise, choose which scenario you want to work on, discuss what it is that you would recommend to the two patients that are involved, and we'll bring you back after about 15 minutes of discussions to get some feedback.
Durhan, are you here?
Yeah, I'm here.
Okay. Did you see Tanya's message?
Oh,
She's not able to lead.
okay. Who else is in Tanya's group?
Let's see. 1 second. Just have to remember what group she was in. 1 second. She was in group 8. So Ida, Bernadette, Carla,
Ida's
Roberta.
Ida was
Yes. Says not joined actually. So maybe she's left. Yeah, they only have 6 people in that room. So 1, 2.
Who's in there? You know?
They're down to 4. So Carla,
There's only 4 people in that group.
Zadie and Xian. I'm not sure how
Can
to
we move
pronounce
them
that.
to another group?
Sorry?
Move those people into another group.
All right,
Just dump them into another group. How's that?
sure.
Under,
Yeah, I'm just bumping them around.
there's still some people on this. I see as participants. Some people not moved.
No, you're the only one that's not assigned.
Well, I
It's
still
just you.
see people listed on the list here.
What do you mean you see them on the list
If I look at
that say not joined?
it just says participant.
If it says not joined, it's because they have left the meeting or they,
It just shows them on my participant list.
they're on your participant list?
Well, I don't know. If I look at the screen, it says participants.
Yes. And it says 9, okay. Um, Yeah, these are the people that have either left the meeting
There's some
or
people
have
late.
refused to join. I can't really do anything with them. There's 9 people here. I can't
Were
move
they
them.
assigned to a group?
Sorry?
Were they assigned to a group? So some people joined the forum late. Have to apologize terribly. We have meant to have a lot more time for the exercise and a lot more time for feedback. And unfortunately, we got ourselves, I think, quite far behind, but hopefully the longer than I had intended initial introduction to the gene therapy was really something that people found worthwhile. I think there was a lot to it. So I'm just going to ask you very quickly, given that we only have about 10 minutes, and I'd love to get some quick reflections from people. Any of the reflections could be on the exercise, but also kind of overall where you're thinking now in terms of gene therapy. Is this, something that you can see as immediately impactful and available for people? What about the case studies that we gave you? Does this give you any terms of thinking about whether or not when people should get access? So I'm just going to ask some of you who are maybe the leads for the groups to maybe give us a quick feedback. And I'm kind of looking at Monica, so I might actually turn to you, Monica. And number one, a huge salute to Monica, who's been hanging in with us from Australia. It must be like 2 o'clock in the morning. I really thank you so much. Monica, maybe I'll ask you for some quick feedback.
Thanks, Duhan. I hope I can still make sense at this time of the morning. We had a great group and we reflected on the need for both advocacy and the value of becoming involved in clinical trials, just because of the quality of care that generally is associated with the clinical trial, and particularly over time. We did spend a bit of time introducing ourselves as well, so that ate into some of our time.
Okay, good. Did you come to any kind of conclusions as to whether or not you would take a therapy that's available now, or should you kind of think about clinical trials?
I'm not sure we landed on an answer to that, but I think that the general kind of feeling is that families will do whatever is available.
Okay, good. Thank you. Okay, somebody else that can actually give us some quick feedback here. Don't be shy. Ada, maybe I'll turn to you.
Hello, everyone. So I hope that it was okay. We made a choice to focus on scenario 2, and it seemed to be an agreement amongst everyone in the group that they feel like Lizzie should start on gene therapy, whereas Ken should be on injection therapy because he was older. And if it's discovered that the gene therapy works, then they would Ken would get the option of starting gene therapy. The very interesting thing that came forward was actually in terms of counseling the family, and it kind of came forward that even if gene therapy is available on compassionate access and clinical trials, most countries do not have access to it, not even high-income countries. So in the group, there were people from areas where there isn't any clinical trials or compassionate access. So they absolutely cannot apply for gene therapy. but there were also people from countries where there are access, but they cannot access it because it's extremely challenging to get into these programs and the clinical trials. So I think that was a very interesting aspect of the discussions, that we're discussing these scenarios, but most of the people with a rare disease actually do not have access to these gene therapies.
Okay, interesting. All right. So at this point, it still is too far removed in terms of the capacity in many of the countries. Even if there was a compassionate access, it wouldn't be really available. So, okay. Somebody else that would like to report back on what their group talked about and any conclusions or not. Russell, is that you volunteering?
Yeah, shall I quickly jump in? We looked at the first one, obviously, for those that know me anyway. Elise was with us, so we were fortunate in having her input. And of course, we grilled her a little on the further background to the alternative treatments that might be in the pipeline. My take on things were that the choice is going to have to be to wait whatever happens, and therefore my recommendation would be to counsel for approval of the existing treatment, even if the trial might be better suited to the particular patient, because that action is at least something they can be doing, and it will hopefully help some people. And we don't know, of course, if the pipeline products might come to anything. Simona mentioned very strongly the need for a much better natural history in order to make decisions on these things, and I think everyone agreed with that. And Danielle questioned, and this is a difficult one, but the ethical rationale for such hugely expensive treatments. And we could spend, let's face it, a whole afternoon discussing that alone. And I certainly have some understanding of that, but no answers. Thanks.
Yeah. And we did not, of course, we deliberately chose not to wander into the cost of the therapies, but that is, in fact, one of the huge, huge issues that everybody has to come to grips with. So, you know, very important. Okay, anybody else that would like to kind of wonder? Mary, yeah.
Yes, so good afternoon, everybody. So our group chose the first scenario. So we looked at, we have two cases there, two patient cases there. So for the first case, our group think that because of the severity of Max's condition, we chose the first option, which is apply and advocate for AAV gene therapy because of the severity. Then for the second scenario, we chose the second option, but something that even though it is moderate, they should still apply for the AAV gene therapy. And for the counseling issue, we think that yes, finances are crucial, especially for low and middle income countries where we don't even have these therapies there. So counseling is crucial in terms of finances, in terms of side effect that they should expect, so that as they go through it, they don't become no surprises for them. Yes, so this is what we think.
Thank you. Thank you very much. And I really look forward to also your participation, because our next workshop is going to be on gene editing, which I think is one of the areas where sickle cell disease is going to be looking very closely in terms of available therapy. So thank you. Okay, somebody else would like to feedback there. Nicola?
Sure. Hi, everyone. I'll try to make it quick. We ran out of time, so we didn't get to talk much about the counseling piece, but the piece that was very evident was that everyone chose the gene therapy that, you know, and we focused on the second one, which was the SMA sort of example. And despite the fact that there was an option for injection, everyone agreed that gene therapy would be what they would want to get them on, regardless of where they are in the disease. The only issue there was not understanding whether they would qualify for the clinical trial. So they would want to look at whether or not they would qualify for the clinical trial, whether there are any immunogenicity issues getting on the gene therapy, and also we don't know whether they would qualify for compassionate use. And so we decided that it would make sense that the injection therapy, if they aren't able to get onto the gene therapy right away, or there's time delay getting into gene therapy, that the injection therapy would make sense to start until they can qualify or get into a gene therapy program or get compassionate use.
Great. So, I mean, very much, I think, reality there, but certainly the preference would be gene therapy. But if there's going to be any delay, let's make sure we get that child started. Fine. Okay. So, Ronnie, can I get you to jump in here quickly? We're just about out of time here.
Oh yeah, I tried to put something in the chat just in case, but we had such a wonderful conversation because there were people from 3 continents on our call, and the issue of access came up, and the role that RDI and patient organizations can play in pushing for access, and especially in low and middle income countries, was central to our discussion. Thank you.
Okay, thank you. Well, certainly the access issue, I think, as even Russell was beginning to talk about as well. Anybody that didn't get a chance to report back, any other quick shout outs I can get here? Because I know people are going to drop off. Okay, I really apologize that we ended up running so tight up against the deadline. Obviously, we do not have these things working in the timely fashion that we had hoped to, but it's so much appreciated. First of all, of course, our 2 speakers, Dr. Ayan just left, and absolutely everybody for your engagement, for your input into the chat line. We will do a follow-up survey with you and hopefully, be able to get some feedback that will allow us to move forward in terms of our next session, which I think will be equally exciting, some different issues with regard to gene editing, as you heard about today. And we will send you out certainly some materials ahead of time, and look forward to seeing you the next time. If you've got any other comments or questions, be sure and let us know. Swani, maybe I'll turn it back to you. Anything else from RDI's perspective in terms of people's feedback or other connections here?
I think the need for this discussion in RDI level was certainly clear. Everyone in the group said it's great that we've been having these conversations at Eurodis, at CORD, but that's a different quality of conversation. And the issue of advocacy and access was completely central to our discussion, and also the agency of the patient community. So I think we're looking forward to workshops 2 and 3 that will look at different frameworks for access, the ethics around it, the WHO's role in this. And I just want to thank CORD for initiating this first patient RDI member-led workshop.
Well, I thank everybody, and thank you, RDI, for partnering on it. Well, as I'm looking at this, and yes, definitely we'll make the presentations available to you, no problem on that, as a slide deck as well. But I'm thinking, we might want to think about session 3, because we haven't taped it yet, and we might actually want to use this, Ronnie, for focusing on access issues, as opposed to moving into, I was going to talk about the choices, if you've got a therapy when there's already other options, but maybe we should really focus on, what are the challenges around access, and certainly much more global access. So let's think about it. We will canvas the group and see if in fact we should be moving much more in that direction. So huge thanks to everybody for participating. You're wonderful. We'll look forward to our next meeting. And I really have to give Monica a shout out for perseverance. God bless you. Goodbye, good morning, good night, where everybody needs to go. Thank you so much.